Hi, my name is Dan, and I'm a fellow in the IGERT in Comparative Genomics at the University of Arizona. I'm also a PhD student in the Department of Computer Science, where I study protein multiple sequence alignment. New sequencing technology has made it possible to use genomic data for many projects across biology, from personalized medicine to the resolution of the tree of life, or even looking at horizontal gene transfer in microorganisms. Even with the diversity of projects, they all follow the same basic process. It starts by sampling the organisms of interest. This is then sent to a sequencer. This machine forms the barrier between the wet lab and the computers. This new data is aligned in some manner, either with itself or with other known sequences. It is only after alignment that biological analysis can be performed. My research focuses on the step of sequence alignment, in particular, the alignment of multiple sequences of proteins. While integral to almost all sequence-based biology, the subtleties of changing this sequence alignment step can sometimes be overlooked. I'll start by explaining what I mean when I say multiple sequence alignment. Assume we have the three sequences shown, and that each one came from a different sample of a protein that performs the same function. What an aligner tries to accomplish is to align the amino acids, represented here as single characters, so that similar letters are in the same column, without changing the order of the letters in each sequence. What we end up with is a pattern of insertions and deletions in each string, represented here by dashes. This pattern often helps us determine the evolutionary history of these sequences or the organisms from which they came. This is not the only pattern of insertions and deletions that accomplishes our goal of aligning similar characters together. The question that we have is what, if anything, makes this choice better than others? Standard aligners make this choice based on user-specified parameters, but many times these parameters are not changed from the ones suggested by the program designers. While the default parameters work well in general, they may not provide the most accurate results for some alignments. What my line of work tries to determine is which alignments are best. If we can do this, we can make a better parameterless aligner. Here's how it works. We start with an accuracy estimator. We call ours Facet, for Feature-Based Accuracy Estimator, which uses simple alignment features as measures of quality. The features are based on what we already know about good alignments. We then use a weighted combination of these features to compile a finalized accuracy estimate. To use this estimator to find a better alignment, we need to have good alignments to choose from. We use several complementary alignment parameters to produce candidate alignments. We then output the candidate alignment with the best Facet score. We call this entire process advising. Using advising and increasing the number of candidate alignments, we greatly improve the accuracy of alignments chosen for analysis. You can see that even with just 5 parameters, you get a significant improvement. For more information, see our poster, or visit our website for software downloads and an expanded version of this video.